Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the isotypes of the antibodies. From the last couple of videos, we are focusing on the immunoglobulin G and its different subtypes. And we have already discussed in detail about the first three subtypes of the immunoglobulin G, which are the immunoglobulin G1, the immunoglobulin G2, and the immunoglobulin G3. And in this particular video, I want to focus on the last subtype of the immunoglobulin G, which is known as the immunoglobulin G4. If you talk about the immunoglobulin G4, they are the least abundant of the subclasses of the immunoglobulin G, and they are only making about 4 to 6% of the total population of the immunoglobulin G in the body. If you talk about their glycosylation pattern, so just like the other subclasses of the immunoglobulin G, they are having this biantennary complex glycan attached at position number 297. Now, if you talk about this biantennary complex, so let me repeat this for you. This is antennary. That means you are going to see antenna-like structure. This is one antenna-like structure. This is another antenna-like structure. Therefore, we call them is the biantennary because this bi, it refers to two. So you are going to see these two antenna-like structure when you talk about the complex glycans that has been attached to the immunoglobulin G4. Now, in this complex glycan, you are going to see different kind of the materials, the molecules. For example, you are going to see the sialic acid, and that is generally moderate in immunoglobulin G4 when you compare that with the other subtypes of the immunoglobulin G. And this is actually responsible for its anti-inflammatory properties. These are the sialic acid that you can see in the biintensity complex glycan structure of the immunoglobulin G4. Now, the galactose level, they are moderate to high in the immunoglobulin G4, and these are actually the galactose structure, as you can see over here. And the third thing about them is that the level of the fucose that tends to be low in the immunoglobulin G4, and these red one, they are actually making the core fucose present in the biintensity complex glycan structure. Another important property of the immunoglobulin G4 is they have got a very short hinge region, which is about 12 amino acids in the human being. So when you talk about the immunoglobulin G4 of the human beings, you are going to see the short hinge region about 12 amino acid long. When you talk about their biological properties, the biological properties of the immunoglobulin G4, so they are the very weak activator of the classical complement pathway. And the reason for their weak activation of the classical complement pathway is they have got a very short hinge region that just I told you. And this short hinge region is actually not providing an optimal binding site for the C1q, and the C1q is the first molecule responsible for the activation of the classical complement pathway. So if the immunoglobulin G4 cannot interact with they cannot interact with the C1q, that means that is going to be a weak activator of the classical complement pathway. Another important property is that they are very a weak activator of the antibody dependent cellular cytotoxicity. And the reason is, again, they have got a very short hinge region, and this short hinge region is going to make them less flexible, and this less flexibility is going to make them a very weak activator of the antibody dependent cellular cytotoxicity. Now, in my last videos, I've told you that the lower fucosylation that is responsible for enhancing a strong interaction with the Fc gamma receptor. But when you look at the overall structure of the immunoglobulin G4, like its short hinge region, its less flexibility, that is going to collectively make the immunoglobulin G4 a weak activator of the antibody dependent cellular cytotoxicity. So this is favoring the a strong ADCC activity, but the short hinge region, it's less flexibility. So when you look at the overall scenario, the immunoglobulin G4 is a weak activator of the antibody dependent cellular cytotoxicity. Now, the placental transfer of the immunoglobulin G4, that is not that much efficient as compared to the immunoglobulin G1, and there are reasons for this. When you talk about the weak placental transfer of the immunoglobulin G4, usually certain substitutions that are responsible for them. For example, if you talk about the position number 311 in the constant domain of the immunoglobulin G4, so in the IgG1, the immunoglobulin G1, you are going to see a glutamine, but in the immunoglobulin G4, you are going to see a proline. Secondly, if you talk about the position number 332 of the CH2 domain of the immunoglobulins, so in the immunoglobulin G1, there is a histidine, but in the immunoglobulin G4, there are serine. So you are going to see some substitution between the immunoglobulin G1 and the immunoglobulin G4. And these substitutions, they are actually making the immunoglobulin G4's reduced affinity for the neonatal receptors and the neonatal Fc receptors. And in my last video, I've told you that the interaction with the neonatal Fc receptor of an immunoglobulin is actually making its transfer through the placenta from the mother into the fetus. But as there is reduced affinity of the immunoglobulin G4 for these neonatal Fc receptors, the immunoglobulin G1 have got a very strong affinity for these neonatal Fc receptor, and hence they are efficiently transferred. But because of the substitution, the immunoglobulin G4 have got a reduced affinity for the neonatal Fc receptor, and hence an inefficient transfer through the placenta. So in the next video, I'll be talking about the another important type of the immunoglobulin, and that is going to be the immunoglobulin A.